All right, so we're back with the super duper speed round through this. A lot of this is redundant. You could watch the first full review, and I would probably go over this in more detail and more slowly. But if you just want to watch this one once, tonight or tomorrow before the exam, it has the kidney and the digestion stuff at the end in kind of the short fashion. So super fast. This is just going to be things you need to know for sure off every slide. Obviously, Fick's law of diffusion, it's important in the lung at the pulmonary capillaries and at the tissue, so you just need to know that simple equation down at the bottom. For pulmonary surfactant, you have to know it reduces the work of breathing. You have to know the diseases associated with it, which is respiratory distress syndrome in either newborns or the elderly. The obstructive and restrictive lung diseases, the only change to this is since the lab TAs are teaching you that edema is technically a restrictive disease, that's not what I was taught, and there are sort of arguments for both. The fluid edema portion, water is obstructive, but the causes of edema, which is pressure in the pulmonary capillaries, is restrictive. So you can know that it's both, or I basically won't ask a question that's gonna, have you choose between that. But you still need to know obstructive and restrictive diseases. This is the summary of the PO2 and PCO2. It doesn't have all of the detail in the lung, so this is really just showing you the blood values more so than the lung values. But it's real simple. Everything on the left in the blue is the same. Everything on the right in the red is the same. And then the alveoli, for what it's worth, is coming into equilibrium with the pulmonary veins, so those numbers are almost the same. So there are more than these that you have to know, but here it is for most of it. Okay. Here is the zoom in on the lungs, and remember the only difference in the lungs here is that because there's dead space, there's differences in inspired air, alveolar air, and expired air. So those numbers vary in those locations. The hemoglobin dissociation curve, you should know where PO2 is for arterial and venous blood, so 140, and you should know how many hemoglobin bind at 25, 50, 75, and 100% saturation, which is 1, 2, 3, or 4. This is summarizing the transport of oxygen and CO2 in the blood. Hemoglobin holds both, and including hydrogen ion and things like cyanide and carbon monoxide and all kinds of stuff. But oxygen is either dissolved in plasma or, by and large, it's bound to hemoglobin. So like 99% is bound to hemoglobin. Carbon dioxide can be in lots of different things. It can be dissolved in the plasma, it can be dissolved in the cytoplasm, it can be dissolved and then dissociated into hydrogen ion, and it can bind directly to hemoglobin. So it just is sort of making note of all the different locations that it is. The other thing that I'll say about this is hemoglobin shifts from right to left. So in the lungs, it's left shifted, and in the tissues, it's right shifted. Remember that affects the affinity of the binding of oxygen to it. And then as carbon dioxide kind of piles on in the tissues, that's the Bohr shift, and that helps to deliver oxygen to the tissues. Okay. All right. The control of ventilation mostly goes because of carbon dioxide, and so this is showing you the sensitivity of your breathing rate to carbon dioxide. The more carbon dioxide you have in the blood, the more you will breathe to get rid of it. It's sensed or can be sensed in the carotid arches, but most of it is being sensed in the brain because on the right hand side here, it dissolves into carbon dioxide and hydrogen ion, and the brain responds to that hydrogen ion. So it is sensed in two locations, but it's mostly the brain that is driving your breathing rate. We didn't really stress this. There are acid factors that will make you breathe, like lactate, and you compensate for it by ventilating more to get rid of CO2. And then last but not least, this was the direct effect of really low oxygen on breathing rate. It goes up only when the oxygen is almost half what it should be. So it's not very sensitive until it's almost a life-saving thing. And this just summarizes all of the different things that affect ventilatory control. All right, for exercise, make sure you know perfusion and diffusion limited. I'm not going to go through all the differences. I think we did that pretty well in class. For exercise physiology, you should know two different things, rest to maximal and then training effects. Obviously know the VO2 max curve, like what the axes are. Obviously know that heart rate goes in linear fashion with VO2 max. Um, this is showing training effects, but also for the first time the anaerobic metabolism as it accumulates. And so both VO2 and anaerobic metabolism are sensitive to training. You can improve your performance for both of those. This was minute ventilation, arterial oxygen, arterial CO2, and hydrogen ion as you approach maximal exercise, and make sure you know what's going on in this. This is the hyperventilation that happens with almost everyone near maximal exercise. Everyone breathes harder. People who are trained tend to delay that as much as possible. Oxygen debt, muscle fatigue, which is review, but mostly so that going into if you bonk or overheat or run out of substrate, you remember how it works with exercise. Hydration is important because you overheat and your heart takes up the extra slack. Overheating can be lethal, so if the brain knows that you are very close to overheating, it will try and shut down the muscle. So that's why Gabrielle Schleiss, whatever her last name is, was, her left side muscles are basically weak because her brain is trying to get her to stop exercising there. Substrate stuff, remember, you need it for muscle contraction, muscle relaxation, and nerve action potentials. This is all review from muscle. This is kind of review from muscle, but uh, as you get more intense with your exercise, fat is great. You have lots of fat, but it's just kind of too low as a throughput for energy. So it will give you some energy, but it's just not enough for the demands that you're asking at maximum exercise. So there's a reliance instead on carbohydrates, which is great in that you can get a really high production of energy, but only if you have sugar. And it's hard to run out of fat in the body. It's very easy to deplete your sugar, and then you bulk. Gender differences, this is easy. VO2 max differences change with aging. Your heart rate goes down, your VO2 max goes down. Body composition stuff, gender differences, all the things that make women more or less powerful and slower in almost every event except for long distance swimming. The effect of detraining happens very quickly. It takes a long time to retrain. Most of what's happening in detraining is the heart is getting smaller, therefore your stroke volume goes away, your cardiac output, that's what Q is, goes away, and as a consequence, your resting heart rate goes up. So having high heart rates at rest is not showing that you're trained. Having low heart rates is demonstration of training instead. Distribution of blood flow, maximum cardiac output goes up, as does the delivery to the muscles. Almost everything else gets turned down. Almost a constant supply to the brain, and greater amount of blood going to the heart. Training effects from neurological things and training effects from steroids. Limits to exercise, genetics, your heart really are the two biggest things, and your blood. Most people don't have issues with their lungs being a limiting factor. Almost no one has that problem. Okay, reproductive stuff. For the male, the anatomy, the epididymis, the vas deferens, male accessory glands, know what comes out of them at their locations and how they improve fertility. Female reproductive system, the anatomy, eggs come from ovaries, fertilization in uterine tubes, implantation in the uterus. The ovarian cycle, first part is follicular, second part is luteal. I fixed the slide on this. 13 days back from the onset of menstruation is when ovulation happens. That's the new, latest, greatest data. These are the hormones that influence ovulation. So LH, FSH, estrogen all spike. Progesterone comes on later. Both estrogen and progesterone go away if there's no pregnancy. They stay on dramatically if there is pregnancy. So here's the ovarian release of an egg up to ovulation. Here's the thickness of the endometrial lining throughout a menstrual cycle. Unfortunately, this one's on a 28-day cycle. Here is everything on in this case, it's a 33-day cycle. Follicular phase first, variable. Ovulation lasts for 24 hours. Sperm can live for 5 days. Luteal phase is 13 days and takes you out. Okay, remember the fimbriae are the things that collect the egg when it's released from the ovary. Fertilization happens in the ampulla. Multiple sperm, maybe 1,000 or so, will cluster around the egg. Hopefully only one at a time gets in, and then HCG, human chorionic gonadotropin, alerts the body that the egg is fertilized so that you don't start a new menstrual cycle. Oral contraceptives, know what they do. And then we're into hormone stuff. These are the different ways that you can have cell-to-cell communication in the body. Paracrine, neurotransmitter, hormonal, neurohormone. Hormone functions. If it targets an endocrine gland, it is a tropic hormone. If it does not, it is a non-tropic hormone. The chemical classification, most things are protein or peptide based. A few are steroid based. So these are just kind of examples, but not 100% complete. Pineal gland makes melatonin. Know what it does. This is the hypothalamus and the posterior pituitary, which makes ADH and oxytocin and releases it. This is the hypothalamus and the anterior pituitary, which have a relationship where the hypothalamus controls, by releasing factors, the anterior pituitary hormones through this portal system of very, very short blood supply, so it happens very quickly. All of the hypothalamic hormones that control the pituitary hormones, what the targets of the pituitary hormones are, and anything else that comes out of those third level stuff. So everything on here you should know. Okay, pancreas involved in blood sugar control. Here you're going to see it again in digestion, but here glucagon and insulin work against each other. Glucagon promotes blood sugar, insulin generally removes blood sugar. Diabetes mellitus has, that went away, 3 hallmarks: polyuria, polydipsia, polyphagia. So that's high urine flow, high thirst, high hunger. And it's back. Here's the 2 different types. Type 1 is autoimmune. Type 2 is the cells stop reacting to insulin, whereas in type 1 you don't make insulin. So type 2, diet and exercise are fixed. Type 1 you generally have to treat with exogenous insulin. Into the digestive system, all the different organs in a row. This isn't that hard, tube in a tube. Secretions that come out of things, this is exocrine, so this is not hormones, this is like ions, mucus, enzymes, things like that. The stomach has all these different cells. Each cell secretes a different compound that is important. So mucus for protection. The parietal cells secrete acid and intrinsic factor for vitamin absorption. And the chief cells secrete pepsinogen, which is the precursor for pepsin, which you use for protein digestion. Hydrochloric acid is important for pH, break down the proteins, and kill bacteria and things like that. Here's pepsin. It comes as pepsinogen, the acid converts it, and then the pepsin will begin breaking down proteins into peptides in the stomach. Okay, the dangers of hydrochloric acid and pepsin, both of these things are corrosive, so they will destroy the stomach unless you protect against them with mucus and not turning on pepsin until you have something in the stomach. Digestion and absorption in the stomach, the only things that go across it are alcohol and NSAIDs, which is aspirin or ibuprofen, and almost nothing else. So not even water is mostly absorbed in the stomach. That's all in the small intestine. You can have peptic ulcers. It could be bacterial. It could be from too much acid. Absorption of the substrates, carbohydrates, all these things need to be broken down except for cellulose. You cannot break it down. You can only absorb glucose, fructose, and galactose, which are monosaccharides. Proteins have to be broken down first by pepsin, then by trypsin and chymotrypsin. So pepsin in the stomach, trypsin and chymotrypsin in the small intestine. And then these were the special enzymes that broke off terminal free amino acids at the end. And there is a table which shows you where all this stuff comes from. So it's not in this one, but you can look it up in the original one. Fat digestion, bile salts comes from the bile glands and the liver. the enzymes lipase break them down. You finally get them down into free fatty acids, and it's absorbed not into the blood, but into the, they call them lacteals here, but you know them as the lymphatic system. Okay, vitamins, easy, just know those. Here's the pancreatic enzymes. Some of these are for proteins, some of these are for sugars, some of these are for nucleic acids. Things that the liver does. All right, to the kidney. The kidney has these two different types of nephrons. That's the functional unit. So the long one is juxtamedullary, the short one is cortical. There are multiple capillaries. So the capillaries that filter the water from the blood into the nephron in the first place are called the glomerulus. Then you reabsorb water, that's called the peritubular capillaries, and then the vasorecta are for random concentration of urine, which we didn't have time to talk about. Regions of the nephron, they have names. So Bowman's capsule collects the fluid from filtration. Proximal tubule is for reabsorption of water and sugar. That's really important. That happens right there. Distal tubule is for reabsorption of a lot of other things, and the collecting duct is where you tweak the final concentration of urine before letting it go to the bladder. And that's where ADH acts, antidiuretic hormone. Here with the capillaries, glomerulus, peritubular. For the juxtaglomerular ones, they have the vasorecta. The Bowman's capsule is important for filtration. So at this point, the blood is pushing water out at high pressure, and you need to know the three different layers. So there are holes in the capillaries that let stuff out. Then there is the basement membrane, which screens proteins. It has charge on it. And then there are podocytes, which screen things like cells from going through. So just know all that stuff. Okay, all that stuff too. There are pressures, just like in other capillaries. So the Starling forces, which control filtration amounts and hydrostatic pressure, proteins staying in the blood to pull water back, and then just having water in the Bowman's capsule. So just know those pressures. The concept of renal clearance, stuff that is filtered into the nephron can be reabsorbed. You can secrete extra stuff. You can kind of leave it alone. You have all different types of options. So I guess I meant to have another slide in there, but things to know: glucose generally has a clearance of 0. You reabsorb all of it. Things like hydrogen ion would have a variable clearance. You can let some go, or you can hold on to it depending on pH. All right, here is the simple version of how antidiuretic hormone, which says vasopressin, but it should say ADH right there instead. It's awesome. This puts aquaporins into the kidney so that you can reabsorb water back. So this concentrates your urine. It is antidiuretic. Remember the things that interfere with ADH: alcohol interferes with the release of it, and caffeine somehow messes with its action in the nephron, but we don't understand how.